Hello students, today in this video session we are going to study about the most fascinating organism on this earth, that is about the bacteria. So before going in detail, let us see where actually the bacteria is present. So bacteria is called ubiquitous, or it is called cosmopolitan in its distribution. The reason for this is the bacteria is found everywhere in all the possible environmental conditions, even in the extreme environmental condition also. So as we are observing here, it is present in the hot spring, it is present in highly acidic soil, even it is present in very low temperature, and it is present even at the depth of the sea also. So this is the cosmopolitan distribution of the bacteria. So it is present everywhere where actually other living organisms cannot survive. There are few reasons why bacteria is having this ability of cosmopolitan distribution. The very important reason for this is the extremely small size of the bacteria. So within a given space, within a given area, more bacterial cells can accommodate. If one organism fails to survive, then other organisms will be there which will continue reproduction and it will continue its generation. So extremely small size is one of the very important reason for its cosmopolitan distribution. Another reason is that the bacterial nutritional mode is highly diverse. So it is having different types of nutrition. One or the other way, it will get the nutrients and it will survive. The third very important reason is its rapid multiplication rate. If all the environmental conditions are favorable for bacteria, it can reproduce, it can produce the offspring only within 20 minutes. So this is the rapid rate of multiplication which is not possible for any other living organism on this earth. And last but not the least, the reason is during the unfavorable environmental condition, bacteria produces the perinatal structure called endospore. So this endospore can survive for number of years, and upon getting the favorable environment, it will produce the new bacteria. So these are the very important reasons why we can find bacteria everywhere, in every possible environmental condition. So I can say that there is no place on the earth which is devoid of bacteria except the burning fire. So this is the wide distribution of the bacteria. Let us see the size and the shape of the bacteria, or altogether I can call it as morphology. So we know that the bacteria are the microorganisms. They cannot be visualized through the naked eye because of its extremely small size. So it is ranging in micrometer. Therefore, in order to visualize the shape and size of the bacteria, we need certain instrument, that is none other than the microscope. So whatever the compound microscope we use in our academic laboratory, its magnification is not enough to study the detailed structure of the bacteria. What we can do is, just we can see the size and the shape of the bacteria with the help of compound microscope, which is called morphology. So in order to study the detailed aspect, which is called ultrastructure of the bacteria, we need to have the higher magnification which is possible in electron microscope. So we will see both morphological features of the bacteria, that is the shape and the size of the bacteria, and even the ultrastructure of the bacteria in a very brief manner. The first shape in which bacteria appear is the spherical shape. If the bacterial cell is there in a spherical shape or it is a round shape, then it is called cocci. So cocci is a round shaped bacteria. If a single spherical shaped cell is there, then we will call such bacteria as So you can understand the meaning here. Mono means single and coccus means spherical shaped cell. So single spherical shaped cell, if it is there, then it is called monococcus. Then the second one we are observing here, two spherical shaped cells are there together forming a bacteria. Such bacteria is known as diplo, means two, coccus. Okay, so two spherical shaped cells, if it is there, then it is termed as diplococcus. Then, if more than two spherical shaped cells are there together, if they are present in the form of a long chain, then it is said to be streptococcus. So streptococcus is nothing but more than two spherical shaped cells which are present in the form of a chain. Okay? Then if more than two spherical shaped cells, if they are present in a group, in a bunch, okay, then it is named as staphylococcus. So these are the spherical shaped cells more than two in number which are present in a group. Then it is staphylococcus. So these are the different shapes, these are the different forms of bacteria if they are present in spherical shape. Similarly, the bacteria may also be present in the form of a rod. Rod-shaped bacteria are also there in nature. Rod-shaped bacteria are called bacillus. So bacillus is the name given to the rod-shaped bacteria. There are different forms in the bacillus also, as in case of coccus. Let us see that if two rod-shaped cells are there together, then it is called diplobacillus. So you can apply the same logic here. Diplo means two, bacillus means rod-shaped cells. So two rod-shaped cells are present together, then it is called diplobacillus. If more than two rod-shaped cells are there which are arranged in the form of a chain, then we can call that bacteria as streptobacillus. So strepto means chain and bacillus means the rod-shaped cells. It is called streptobacillus. So these are the different forms which occur in the rod-shaped bacteria. Other than this spherical and rod-shaped cells, bacteria may also be present in the form of a comma. Comma-shaped bacteria is also there, which is named as Vibrio. So Vibrio means comma-shaped bacteria. Other than this, bacteria may also exist in the form of the spring. If the bacterial shape is spring-like, then it is called spirulum. So these are the different morphological shapes of the bacteria. So if we think about the size of the bacteria, based upon the shape, the size will also vary. In general, we can tell that bacteria is 0.1 to 1 micrometer in its size. So this is the normal range. Of course, there is no rule that all the bacteria should have 0.1 to 1 micrometer in its size. There is lot of variation, but this is the general size what we can tell. So among them, the spirulum, if the bacteria is there in spring shape, then it will attain the maximum size. Okay? So this is about the morphology of the bacteria, whatever we can see with the help of compound microscope. Now, so here we are observing the size. Of course, we are comparing the size of the bacteria with the other cells. So if you see the size of the atom, then it will be there in the nanometer range. If you see the other component biomolecules, like lipid and the protein, they are also there in the nanometer range. If you see the bacteria, it will fall somewhere here in the 1 micrometer range is there. So which is similar to the size of the mitochondria, the very important eukaryotic cell organelle, as well as the flu. If you compare the size of the virus to the bacteria, it is much more smaller. Also it is there in the nanometer range. Then on the other hand, if you observe the size of the plant cell and the animal cell, they are much more larger than the bacteria. So this is the general size of the bacterial cell. So it is in micrometer range, right? Now, flagella is one of the very important external appendage which is observed in the most of the bacteria, which is helpful in locomotion of the bacteria for the movement process. So if the bacteria wants to move from one place to another, it will use a very important locomotory structure that is called flagella. So the bacterial cell may have the single flagellum, or it may have more than one flagellum. So many flagella may be there in the bacterial cell. And the amount and distribution of the flagella is also highly variable from one organism to another organism. So based upon this, based upon the number and the distribution of the flagella, we can classify or we can categorize the bacterial cell into different types. Let us see that. If the bacteria is not having any flagella, even a single flagellum is not there, then we can call such bacteria as Heterotrophism. So we can call that bacteria as Heterotrophism. Very simple here. Trichus means hair-like structure, which refers to the flagella. If you add the word before that trichus, then it will indicate that the bacteria is not having any flagella or trichus. Then second one is, we are observing here the bacterial cell and it is having a flagella, single flagellum at any position of the bacterial cell, then it is said to be monotrichous. Very simple. Mono means single, trichous means the hair-like structure, which is nothing but flagella. If a single flagellum is present on the bacterial cell, then it is said to be monotrichous. Then we are observing here the bacterial cell is there and it is having the two flagella is there, which are present on either side of the bacteria, on both the ends of the bacterial cell. Then that bacteria is called Amphitrichus. So very easily you can remember these names. I think you know the meaning of amphi. So you have heard about the organism called amphibia, which is nothing but the organism which can live both in water as well as on land. So there amphi stands for both. So here amphi means both and trichus means the flagella. Flagella is present on both the ends of the cell, then it is called amphitrichus. Okay, so very simple to remember. Then this is the bacterial cell which is having more than two flagella, which are present in a group, which are present in a bundle, and it is attached to one end of the bacterial cell. All the flagella, they are forming one bunch, and that bunch is attached to any one end of the bacterial cell. That is named as cephalotrichus. It is called cephalotrichus. So tuft or bunch of flagella if it is attached to one end of the bacterial cell. Then here we are observing tuft of flagella or group of flagella is there bacterial cell. So here some flagella is there and here some flagella are distributed on both the ends of the cell. That is named as locotrichus. So tuft of flagella, if it is present on both the ends of the bacterial cell, then it is said to be locotrichus. If a single flagellum is present on either side of the bacterial cell, then it is named as amphitrichus. Hope you can differentiate these two, amphi and locotrichus. Okay? Then another condition is there in which flagella is distributed all over the bacterial cell. All over the bacteria, the flagella is randomly distributed. If that is the condition, then that is called peritrichus. Condition is called peritrichus. Usually peri means the surface. On the surface of the bacteria, many flagella, they are distributed randomly. That is the peritrichus condition. So these are the different forms of the bacteria which are based on the flagella distribution. Hope it is clear to you. Now, let us move on to the nutrition in case of bacteria. How it will get the nutrients. So I already told you that the bacteria is having wide range of nutritional mode. Let us see one by one. So bacteria, on the basis of its nutritional mode, it can be classified into two types. The bacteria may be either autotrophic or it may be heterotrophic. The meaning is very simple. So here autotrophism means those bacteria which can synthesize their organic food material by its own without depending upon any other living organism. If it is preparing the organic food material, then it is said to be autotrophic bacteria. If the bacteria wants to prepare its organic food material by its own, then there are two options for the bacteria. It may be either photosynthetic or it may be chemosynthetic. Let us see these two one by one. The first one, photosynthetic bacteria, as the name itself indicating here. We know that the word photon means light and synthesis means the production. So the simple meaning of photosynthetic bacteria is that these are the bacteria which will produce the organic food material by utilizing the sunlight and other components in the environment. Okay? Then it will become photosynthetic. If the bacteria is capable of converting the light energy into the chemical energy. Right? So there are different varieties of photosynthetic bacteria. Two of them, the examples I have given here. The green sulfur bacteria is one example and chlorobium is another bacteria which can carry out photosynthesis. Right? So to carry out photosynthesis, a very important requirement for the bacterial cell is light capturing pigment. So this is very important. If the bacteria is not having any light capturing pigment, then it cannot synthesize the organic food material. Therefore, the photosynthetic bacteria will have a very important light capturing pigment that is called bacteriochlorophyll. Bacteriochlorophyll is the pigment present in the bacteria which enables the bacteria to capture the sunlight and to convert that sunlight into the chemical energy. If the bacteria is not having this bacteriochlorophyll, that is light capturing pigment, then what happens? Let us see. If it is not there, if the bacteria is not having any photosynthetic pigment, then it will go for another mode of autotrophism that is called chemosynthesis. So name itself indicating here, chemo means chemical. Okay, so in this the chemo means chemical and the word synthesis means production. So this is the mode of nutrition in which bacteria produces the organic food material by converting the chemical from one form to another form. Thereby it is getting the required energy. So here the good example for this is nitrosomonas, which can convert the ammonia into nitrate, and nitrobacter, which can convert the nitrate into nitrite. So by converting the chemicals from one form to another form, if the bacteria is obtaining its nutrients, then it is said to be chemosynthetic. Remember, there is no photosynthetic pigment in the chemosynthetic bacteria. Okay? Then, another very important mode of nutrition in the bacteria is the heterotrophism. Heterotrophism means the bacteria is dependent on any other living organism, or it is dependent on some dead components of the environment. It cannot prepare the organic food material by its own. Then it is said to be heterotrophic bacteria. Now here we are observing there are three types of heterotrophic bacteria. The first one is called saprophytic bacteria, or you can also call them as decomposers. So these are the very important organisms on this earth which is responsible for decomposition process occurring on the earth. So these are the bacteria which will secrete certain extracellular enzymes. So those extracellular enzymes act upon the dead organic matter. It may be the dead body of the animal, or it may be the dead body of the plant, and it will convert the complex organic matter into simpler form. So by converting the complex organic matter to the simpler form, if the organism is obtaining the energy, then it is said to be saprophytic bacteria, or you can call them as decomposers. Okay, so this is the saprophytic mode of nutrition. Very good example for this saprophytic bacteria is Pseudomonas. The genus Pseudomonas consisting of many different species which are capable of doing decomposition by secreting certain extracellular enzymes. Right? Then, if the bacteria is not able to produce any extracellular enzyme, then what happens? Then the bacteria will become parasitic. So parasitic mode of nutrition is a special mode of nutrition in which bacteria get its ready-made food from the another living organism. That living organism which is providing the ready-made food to the bacteria is known as host organism. It is called host organism. So whenever the bacteria gets the nutrients from the host organism, one or the other way it will affect the host organism. It may cause the disease to the host organism. Now you may ask the question, which is the organism which can act as a host for bacteria? So host organism may be either the plants or the animals or the human being. Okay, so parasitic bacteria is the one which is obtaining the nutrients from the another living organism, that is host organism. So the next mode of nutrition in bacteria is the symbiotic association. In case of symbiotic association, bacteria will undergo mutual understanding with some other organism. So here we are observing the example of the bacteria called Rhizobium. Rhizobium always associate with the roots of the higher plants. In the roots of the higher plant, it will create a structure called nodule. So here we are observing some knot-like structure which is developed in that is called nodule, inside which the bacterium, that is rhizobium, will take its shelter. Rhizobium produces a very important enzyme. The enzyme is named as nitrogenase enzyme. So with the help of this nitrogenase enzyme, rhizobium is able to fix the atmospheric nitrogen into the root nodules of the plant. So both the organisms are benefited here. As the bacteria is taking shelter within the root nodule, it is getting the ready-made food from the roots of the plant. In turn, by doing the nitrogen fixation, it is enhancing the growth of the plant also. So this is one type of mutual association in which both bacteria as well as the root of the plant is benefited equally. So these are the different modes of nutrition, whatever we can observe in case of